हॅलो सर, युट्यूब लिंक शेअर केली बघा व्हॉट्सअपला. माईक बंद आहे तुमचा.
Hello, not audible.
हा सर, येतं. Yes.
येतो ना आवाज? हा ठीक, थँक्यू. लिंक पण शेअर करतो मी. मी टाकतो ग्रुपला आपल्या त्या वेबिनारच्या. तर सर कोकरे सर जॉईन झाल्यानंतर त्यांना फक्त होस्ट करून टाकतो. मला होस्ट करायची का? कॅन यू जस्ट मेक होस्ट सो दॅट आय कॅन हॅव कंट्रोल.
येस येस. होस्ट नंतर कोणाला करू मी?
नाही, होस्ट मला कर. काय यू कॅन कीप मी होस्ट. आय विल बी हिअर ओनली.
ठीक आहे.
मला
ओके.
होस्ट करून टाक. आय विल मॅनेज, नॉट इश्यू.
हो हो.
येस ना, मी तुम्हाला होस्ट करतो मग. येस, तो पण मी माझी शेअर करतो स्क्रीन. जस्ट लेट मी नो दिस माय स्क्रीन विजिबल ऑर नॉट. माझा आवाज येतो ना व्यवस्थित? हॅलो.
हो येतोय.
हा, मला आता मी एक हे बघ फ्लायर बनवला आहे, ते शेअर करतो. चेक. मी काय करतो, लेट मी शेअर द स्क्रीन टिल दॅट टाइम. दिसते का स्क्रीन? बघते का मला सांग. कॅन यू सी द स्क्रीन?
येस, विजिबल आहे.
विजिबल आहे, ठीक आहे. हेच ठेवतो मग आता. आय कीप दिस
ओके.
स्क्रीन ॲज सच. युट्यूबला बघतो, दिसतं का युट्यूबला? येस, आय कॅन सी. चेंज करतो. ओके, ठीक आहे. So, dear participant, we are going to start a session at sharp 11 o'clock. And also, there's a YouTube live session also going on. Dear participant, we are going to start a session at sharp 11 o'clock. And also, there's a YouTube live. Yes, sir. Progress, sir, I can hear you. Can you see,
Yes,
sir?
yes, sir. Good morning, sir.
Good
Yes, sir.
morning. Hope I am audible to you. You are
Yes,
also
sir.
audible. Yes.
Yes, yes, sir. Yes.
Rename your name.
Yeah.
Yes. Sir, I'll give the right to you for presentation. Just you can check it at your end. I'll make you co-host. Yes.
Then I have to make so just as share slide, no?
Yeah, yes, yes. I'll just मी माझं स्टॉप करतो मग फक्त मला तुमचं चेक करा.
हा म्हणजे मला स्लाइड शेअर करता येते का बघू?
करता येतं बघा ना शेअर मध्ये फक्त जा खाली बघा.
हा. येस.
शेअरला क्लिक करा आणि शेअर युअर स्क्रीन. येस सर.
आलंय ना?
या दिसतंय सर. वी कॅन सी. येत आहे कमिंग. येस आय कॅन सी फॉर युअर 29 स्लाइड दिसते मला. 29 नंबरची.
हो हो.
कम टू फर्स्ट स्लाइड सर. या फर्स्ट स्लाइड्स.
यस सर.
करेक्ट. त्याला असं फक्त हे करायचं म्हणजे प्रेझेंटेशन मोड मध्ये करा. स्लाइड शो मध्ये प्रेझेंट बिगिन स्टार्ट विथ फर्स्ट स्लाइड.
Yes, sir. Yes.
Now we can see sir perfectly.
Perfectly. Yeah.
Okay.
Last time what happens, one or two presentations.
Okay.
From my side, I am not visible, as well as
Okay.
audience also not visible.
No.
Only screen is there. Then it is very
Okay.
difficult to talk
Correct, sir. Correct.
by only screen.
Only screen. Okay. No.
Yeah.
Now perfectly, sir. You're audible. You're visible. Also, we can see your presentation. राहू
Yeah,
द्या सर
yeah.
काय प्रॉब्लेम नाही. नाही सर मला वाटतं तुम्ही नंतर शेअर करा. मला तुमच्या 2 स्लाइड दाखवायच्या आहे इंट्रोडक्शनच्या. नाउ यू कॅन स्टॉप युअर शेअरिंग. मी तुम्हाला जेव्हा सांगेल तेव्हा शेअर करा मग.
Yes, sir.
म्हणजे होत
बघतो
आहे
मला.
म्हणजे होतं म्हणजे दॅट्स जस्ट वी आर चेक टेक्निकल रिक्वायरमेंट.
स्टॉप शेअर करू?
तुम्ही स्टॉप शेअरिंग करा सर.
हा.
आता मी माझी स्लाइड मी शेअर करतो.
Yes, sir.
Yes, sir. I hope you can see my screen, sir.
1 minute, just coming. Yes, sir. Now.
Okay, great.
Yeah.
We'll keep it as such now.
Yeah.
Even sir, आपण YouTube live पण चाललाय म्हणजे YouTube live ला पण आहे ठेवलाय.
हा
काही
हा
पार्टिसिपेंटला याची लिमिट 100ची आहे. तो
हा
काही
हा
पार्टिसिपेंट जॉईन करू शकत नाही, त्यांना युट्यूब लाईव्ह केलाय आपण.
हो हो हो.
And all PhD scholar, sir, MSc, PhD, professor, faculties are joined.
हम्म.
And we got very huge response. There were about 375 participants registered.
टोटल.
Yeah, total registration. We'll start at sharp 11 o'clock. Dr. Arvind Deshmukh sir is joining.
Yes, sir. Yes.
Yeah, I can see on YouTube live also people are joining. Yes. Deshmukh sir.
I think Deshmukh sir is joined. I think.
नाही इथं नाही विचारत. हो दिसतंय त्यांचं नाव.
हो.
सर तुम्ही जॉईन केलंय? जॉईन करा. चालेल. ओके. येस. चालेल. ठीक आहे सर. Yes, sir is joining for another device. Yes. Share. Yes. So screen visible है ना?
Yes, sir. Yes.
Yeah. Yes, sir also joining. I think sir has came or no. Yes, let me check. Dear participants, we are starting a session within 4 or 5 minutes. Please stay tuned. Also, there's a session live going on YouTube channel. So already the link has been shared on your email as well as on your group. We are waiting for President Microbiology Society of India, Sir Arvind Deshmukh sir. Once he joins, then we'll begin the session.
Hello, I have joined, sir. Fine. Thank you, sir.
Thank you. Good morning, sir. Good morning, sir.
Good morning, good morning.
Yes, sir.
Whether Kokre sir has joined?
Yes, sir. Yes, sir.
Okay.
Yes.
So let us wait for 2-3 minutes more.
Yes, Paul,
Because
sir, not audible. Not audible. You.
I am not audible.
Paul Setiwar, sir. Unmute, sir.
Sorry.
Yes,
Sir,
yes.
hope I'm audible now.
Yes. Now it is
Great.
audible.
So now 13 participants are joined. We'll wait for 2-3 minutes. Then probably
Yeah.
I'll
Yes.
with the session.
Yes.
Yes, sir.
More participants will join within
Yeah.
2-3 minutes.
Now coming. As per Indian time, they are joining.
Yes.
Yes, sir. So already I have emailed them also.
Okay.
The YouTube link as well as Zoom link, because we can accommodate over 100 participants over here.
Yes. People are joining. 17.
Yes, sir, joining 7. Yes. I can see people are joining. I'm admitting all now.
Yes.
Dear participant, there's a YouTube live link also I'll share on your email as well as WhatsApp. Those are unable to join, probably you can share that link to your colleagues so that they can also take the benefits of this very important session from our very expert person, Dr. Kokri sir. We begin the session within 2 minutes. So shall we start now so that people will join?
1 minute. Because generally our practice is that we wait for 5 minutes and then we start.
Okay, then we'll wait for next 2 minutes. Now it is 11:03.
Yes.
Good, sir. Quorum will be complete then.
Correct.
Now people are there, relaxed at home because rains are going on. Maybe they can benefit.
Yes.
They can take the benefit of this session.
And it will be recorded on the YouTube also. That
Correct.
Also we are going to circulate.
Correct. That is really great for them. They can watch the session again and they can take the benefits of that. Correct, sir.
Correct.
Sir is joining from which place, Arvind sir?
Arvind sir, I think from home only.
Joining from Pune.
Pune only. Great.
Yes. Yeah.
I came back 4 days back.
Yes, sir.
Yeah.
Yes.
Sir was in US last 2 months.
Yes, sir.
I was in US.
Yes, sir.
And for 15 days I was in Himachal Pradesh and Chandigarh.
Yes,
Great,
sir.
Just before 5 days, I came Pune.
Yes, sir.
Yes. Again, I'm going Kerala.
Great, sir.
Yes.
Then I'm going Jaipur.
Hmm.
You are inspiration for us, sir. You are sharing knowledge. We are lifelong learner. Like you all made us like this.
Thank
And
you. Thank you.
and
Yes.
this is also how the
Yeah.
traveling to different places and all.
Correct.
Yes, yes.
So by mind, he is not retired. By age, might be retired.
Yes. My age is 72,
Correct,
correct?
sir.
Yes, yes.
Great, sir.
But I always say that there is a misplace of 2 and 7. It is a 27.
Yes.
Great thought, sir. Great thought. Yes, dear participant, we are starting session within a minute. I hope you are there
So you can start now.
mentally, physically. So,
You can
yeah,
start.
at the outset, yeah. So we would like to welcome you, all the participants who are joined across the nation. I can see the participant over here. So on behalf of Microbiology Society of India, President Dr. Arvind Deshmukh sir is there on behalf of his and all the team members of Microbiology Society of India. I heartily welcome you all the participants for joining this first webinar series from the Microbiology Society of India and in association with the pharmaceutical microbiology. That's a part of this society. So I really welcome you all the participants, and also I welcome you, our today's guest. We can say not a guest, is a speaker and renowned scientist. Let me first introduce Dr. Arvind Deshmukh sir. He is the president of Microbiology Society of India. Then followed by we'll introduce Dr. Kokre sir. So I first asked Dr. Arvind Deshmukh sir just to begin with what is this Microbiology Society of India? What is the agenda? What is the objective of the society? Sir, I'm just hand over decision to you. Can you just speak your mind?
Thank you. Thank you, Dr. Satish, sir. I'm really happy that Dr. Satish Polshetwar, he has started this lecture series. It is not the only one. And today, inauguration is at the hands of Dr. Kokre, sir. Very renowned scientist. And his lecture will be useful to all students in pharmaceutical microbiology. Again, I am reiterating that pharmaceutical microbiology is a very, very important section of the industrial microbiology. Our Honorable Finance Minister, Honorable Finance Minister Nirmala Sitharaman also made a provision of lots of rupees to boost pharmaceutical microbiology and pharma industry in India. So, it is a great opportunity to the student to learn, and by attending such lectures, they get a more exposure. They will become ready for the interview in various pharma industries. I know Kokre sir from 20 years. He is a really devotee and a dedicated person, as well as highly studied person. So his lecture, his words are useful to the young students. Microbiologist society, I will not talk more on it. I will tell you only that you go on the Google, search website www.microbiosociety.com. You will get full idea, or you go on Google, search microbiologist society, you will get all details of the microbiology society activity. I will not say thanks to Dr. Satish because he is a part and parcel of microbiology society. Only I appreciate his efforts and I wish all the best to this lecture series. Thank you. Thank you very much.
Thank you, Dr. Deshmukh sir. You are always inspiration for us and always the torchbearer for all entire nation, for nation building. I think really thanks. Now it's a time to introduce our today's renowned speaker, one of the great human being I can say, today with us. He has giving time us, and he never say no for us. He always say no is new opportunity for us. So today I'm introduce Dr. Chandrakant Kokre sir. Now recently he's selected as a post. Many aware about that, but that's a great achievement. Now current achievement I can say you. So sir is having a rich experience of 32 years, and he has a vast experience in the academics as well as research. If you look at his H index, more than 49, I-10 index more than 49, H index is 33. Itself reflects the quality and the exemplary scientist. And the research paper he has published more than 92 to all peer review journals. And he published 11 book chapters and 11 books in Nirali Prakashan. One of the favorite book that I know. He's my also teacher. So that is pharmaceutical microbiology. Still, many students will remember him as a microbiologist, and he wrote that first book that is pharmaceutical microbiology, and all the B Farm student, M Farm student referring his books till now. So he's one of the finest scientists, finest author, finest person. So he has a total project, 12 project he has completed from ICMR, AICT, SPPU, industry funded project, total grant more than 38 lakhs. That show how he has been committed to the research and scientific community. Award more than 22 awards. He secured third rank in the selection of Principal Government Pharmacy College MPSC. That recent achievement. Listed in the world top percent scientists. Now recently, Rank Global Registry has come up with this rank, and he has appeared in that 5% scientist rank. I think that's really great achievement, sir. We really salute you for that recent achievement. He listed in top 2% scientists declared by Stanford University. That is also one of the great achievement from 2022 to 2026. Best Teacher Award, I think is the best teacher, best researcher, best scientist, and best human being, I can say. Young Teacher Career Awards in 2005, 2006 from AICT. He has delivered many sessions across the nation in the workshop, national seminar, and conferences. As a resource person, invited lectures. Even he has visited various countries. You can see the list from England to China to Germany to Switzerland to Malaysia to Malaysia. He has published various newspaper articles, membership, fellowship, more than 3. He is qualified MSCIT, approved microbiologist analyst from FDA. That's I think great achievement. So with this all the achievements, sir, I would like to invite you over here to as a speaker, and I'm really thanks to you for accepting my invitation on behalf of Dr. Deshmukh sir, President Microbiology Society of India, and all the team of Microbiology Society of India for this. So now the session is over to you, sir. Thank you once again to all participants also. Yeah, over to you, sir. I stop my sharing.
Yeah.
Yes. You can share the screen, sir, now.
Yes. Yes, sir. Screen is visible now.
Visible, sir. Visible, and you are completely properly audible, sir. Yes. Thank you, sir.
Yes,
You can.
sir. Yes, thanks. Good morning to one and all present for these sessions. First, I am thankful to the President of Microbiology Society, A.M. Deshmukh sir, and one of the young, dynamic, active member of this recently appointed as the President of Pharmaceutical Microbiologist, Professor Satish Sattiwar sir. Today, I am very happy to present myself related to one of the recent topic, that is the bionanotechnology in next generation drug delivery systems. Its audience may be pharmaceutical microbiology. That topic should be such a way that covers some 50% microbiology, some part in the pharmaceutical sciences also. And we have to select such topics that covers both the parts. And based on that, with discussion with the Satish sir, I decided to go for this topic, bionanotechnology, that if audience is microbiology or pharmacy, both are joins together, and that may be applicable to the both areas and both the fields. I'm going to discuss that is how these plants or microbial sources, natural sources we can use for synthesis of the nanoparticles. And everyone knows the importance of nanoparticles in the pharmaceutical sciences or drug delivery systems. I will take some examples, few examples related to the how this nanoparticle is useful or applicable in the system. But basic parts, how are you going to synthesize this nanoparticles that should have the stability, specific size or specific particle size, monodispersity. This is very important parameters if you're going to apply in the biological system. Then what is bionanotechnology? Yes, that is nanotechnology, that is smaller size or smaller components, smaller material science. If you combine with the bio, that is two parameters are there. That is one, how this biological system helps to the production of the nanoscale components. We can use it may be plants, it may be microbial cells, how it is useful to synthesize this nano derivatives. And second, if nanoparticles are there, nano components are there, how it is going to help to the biological systems. That the both ways, sometimes this term alternatively it may be used bionanotechnology or nanobiotechnology. Yes, here it may be used, but it is generally it is a material science and engineering applicable to the biology. And it is not so easy because this technology will require very collaborative research works or collaborative studies that may require the experts of the physics, chemistry, biology, material science, engineering, and for the application that pharmaceuticals that may be interdisciplinary area of this bionanotechnology. Here it is originally what microbiology is there, that is that we're going to synthesize these nanoparticles because microbes are basically microbial source is that how the importance related to the synthesis of the active component as the enzymes, protein, metabolites, and its specific active materials. And we know that very well, that is this microorganism sources, plant sources is the really storehouse of these enzymes, proteins, antibiotics, yes, or it may be have some different applications. Second, that is pharmaceutical part, that is how this particular cell we're going to use to make this formulation easily useful. That formulation is maybe stable based on their specific particle size, that how the specific surface chemistry, as well as they how the specific release profile, because activity of that particular drug molecules or their effects is depends on this all parameters of that particular formulations. And that may we're going to enhance the solubility by using this concept. Yes, microbial cells with the formulations, how are you going to enhance the solubility, tissue targetings, or it may be applicable to the gene delivery, regeneration of the tissue, and that may have some the antimicrobial activities as well as biosensors. And that major parameters, that is biosynthesis, pharma translation, for plant biotechnology is also used because plants is also used for synthesis of nanoparticles. We'll take some examples of this also. And generally we know that they have a lot of methods for synthesis. We'll go into that parts. That is basic biosource, that we can use the microbes, algae, enzymes, DNA, proteins, and that use as a specific plant extracts. And then nano interfaces are there, that is microbial cells not only synthesize nanoparticles, but simultaneously you can synthesize the molecules or protein that we have to make the stable components, and then it may be used for targeting or it may be used for gene transfer activity, have specific applications of these nanoparticles. Why this nanotechnology is most useful in the drug delivery systems? That is, first, we always talk about the targetings of the drugs. Yes, if a drug generally if given by oral route injections, but if has the infection of particular site, their concentration of the drugs should be higher in that particular site. And if it goes to other site, they have caused adverse effects. Hence, targeting is the most important. Second, that is most of drug is class 2 drugs, that is insoluble drugs. And they have a lot of problems if you're going to transfer in the drug delivery systems. Hence, by using this nanotechnology, how are you going to enhance the solubility or increase the solubility? That is again another parameters or one of the most important application of this nanotechnology, that is we can design the specific devices and enhance the solubility. Constant rate of drug delivery, that is again most of diseases are chronic diseases and that require drug to adhere for the longer times or drugs may be present to the particular sites for longer durations, and for that purpose that is drugs should be slowly or constantly released from the devices and should be available more time that in the particular site and increase the drug stability. If it is talking the drug stability, nowadays we are talking to the biosimilars. Always biotech products are higher cost products. Can you synthesize this product by the cheaper rate by using some modifications, some alteration of some methods? Yes, we can synthesize some products, we can derive some products at cheaper rate as the biosimilar products. But most important part in this such type of a product is the stability. And still we are not able to give insulin by any other route. You know the use and application of the insulin as anti-diabetic, we have to give only injections. Yes, most of the vaccines are there in corona type also vaccine. That vaccine is also injections. But some other companies discover the vaccines that useful for intranasal route and that already applied or use this vaccine by the intranasal route. Yes, it is possible this type of the product, we can say high molecular weight products, protein type of the products, peptide type of the products, sensitive products, we can make stable forms and it may be given by specific applications and that may be more useful to the patient by designing drug delivery systems by the help of this nanotechnology. What is the next generation drug delivery system? Lot of drug delivery systems are there. Advanced drug delivery systems are there that nowadays artificial intelligence are coming. Then specific biomolecules or biopolymers, we can biodegradable polymers is also used for designing some nanosciences, some liposomes, polymeric nanoparticles, dendrimers, metallic nanoparticles are there, and some smart or stimuli responsive delivery are there, that is maybe internal triggers or external triggers. Such systems are there, magnetic systems, if you want to go for targeting the drug delivery systems. Even there is the micro robots or biohybrid systems are there that have some applications to use this next generations to treat the some chronic diseases as like cancer, diabetes. Yes, we can use such type of the nowadays. So everyone talking about the artificial intelligence and digital healthcare systems. Paul Setivar is more expert in these topics. Yes, that is such systems we can make more comfortable. We can give lesser dose to the patients, lesser adverse effects, and easy to cure the diseases, such type of the chronic diseases. Yes, we are using wide range of the cells to discover the active molecules. We're going for the recombinant DNA technology. We can going for the biosimilar products, but still we are not able to cure some diseases. Yes, that we have the limitations and still we are not proper treatments. It may be for the HIV, it may be for the cancer, it may be for the diabetes. And that's indicate that we have the more opportunity to go for such advanced system. By using this nanotechnology, is it possible to target the drugs? Lesser amount of the drugs we can target to the particular sites and we can, yes, destruct the cell, or nowadays everyone talking about the gene therapy. Is it possible to replace the cells which synthesize the insulin in the body? That is that cell by the that gene defective cells we can transferred and that cell may be activated to enhance the insulin synthesis in the body. As such studies are going on, but still we are not able to treat such type of the diseases. That is major components or major parts, that is nanoparticles, how are you going to synthesize it? Yes, that is how the specific process. Yes, that is main importance in this nanoparticles, that is getting the right particle size. Generally, we can talking 1 to 100 nanometers. This is the right particle size. We can call it as nanoparticles. Yes, right particle size, making administrable formulations that we can transfer in the formulation form, and that monodispersity, that is most important. Whatever particles synthesized with this process, that have the similar size. This right particle size through self lives, and there is two specific approaches: top down approach and bottom up approach. Top down approach means larger size particles, so we can going to reduce by applying some pressures by milling process, and that particles it may be reduced by the dry milling, wet milling. These techniques we can use, and we can synthesize the specific particle size by using top down approach. Similarly, we have the bottom up approach, that is, it may use specific techniques as controlled precipitations, controlled crystallizations, emulsifications. Yes, this method we can going to use, but still this method have the limitations. All these methods we require a lot of chemicals to synthesize this specific size nanoparticles, require longer times, and this safety parameters is also most important in nanoparticles. Yes, if particles convert into smaller size, 200 nanometer to 100 nanometer to 10 nanometers, their effect or their activity, it may be changed. Yes, hence their effect in particular size, it may be studied. If this particle goes to the tissues, yes, how much time it retain in the tissue, how it is cleared from tissues, how it is distributed, eliminated from the tissues, their cellular and tissue functions, what is their ADME profiles, that is absorption, distribution, metabolism, and elimination profile. Yes, if particle size are different, what is their effects? Particle size is smaller, their effects. Yes, that properly how to study it. If that nanoparticles may targeted in the specific tissues, yes, how it is cleared from that particular tissues, how it is released from the tissues, and if this particle goes to the environments, what is the effects? And animal, fish, plants, yes, that also you studied. Yes, animal, fish, plants. Yes, titanium dioxide, zinc oxide, this component we use in the cosmetic and sunscreens. And if you go for the smaller size, smaller particles, and they have the effect, that is destruction, that may have the DNA damage. Hence, such effects, that is safety parameter, is most important. Is most important, and that how the study before use of this nanoscale component in the pharmaceuticals or in the human being. Then quality aspects, that quality parameters, yes, it may have particle size, size distribution. So how to study it? So what is the size of that particular nanoparticle components? Size distributions, monodispersity, surface area, that ultimately more surface area, that ultimately rate of dissolution is higher, their effect is maybe higher. Surface chemistry is also more important. We have to study surface coatings. Yes, most of times to enhance the stability, there may be used surface coating techniques. Porosity of this particle, surface charge, hydrophilicity, wettability, purity. Again, we are going to use IV routes. Then how the purity, sterility, and stability is again important, because they have the stability. There, this particles, they may have the problem of aggregation, protein absorptions, and they have to study the manufacturing and control parameters. Yes, this is the most important quality aspects of any nano-size components, and you have to study it first. Yes, there is a lot of techniques to use to synthesis nanoparticles. Chemical process, top-down, bottom-up approach. But we have to go another process, that is biological synthesis. Why biological synthesis? Every time we're talking about green chemistry. Yes, similarly, we can use the biological systems. Yes, there is no use of the any chemicals. Yes, you are growing need to develop the environment friendly nanoparticle synthesis. There is no need of any chemicals, and that synthesis of nanoparticles is possible to use specific size, controlled monoviscosity, and they have the specific size. Yes, many researches are there. We're going to take some examples also. Proves that synthesize this nanoparticle from the biological sources, they have the specific compositions as well as specific size. And there is no use of any toxic chemicals in the process of nanoparticle synthesis. And even that problem it is associated with the stability, crystal growth, aggregations, that also avoided. That is, most of the fungal species we're going to use for synthesis of these nanoparticles, that fungal species not only synthesize the particles, but simultaneously synthesize some proteins, and that protein you have to coat these nanoparticles to make more stable. Yes, such a capability of that cell is have to synthesize as well as to make the stability of these nanoparticles. Yes, that is which microbes are used for synthesis of nanoparticles. Many researchers try to use bacteria, actinomycetes, fungi, yeast, wide range of the nanoparticles used for this synthesis of these nanoparticles. This are how the examples of bacteria synthesize intracellular as well as extracellular nanoparticles. Because if bacterial species exposed to some metal ions, expose some metallic compounds, yes, ultimately bacteria have some reflex systems because everyone is trying for the survival. Yes, they have the metal tolerance, and these mechanisms is transferred this component in specific size. Yes, similarly actinomycetes. Yes, we know this species very well. Actinomycetes itself is the bacteria having the some morphology as fungi, and very important group in the pharmaceutical science. Yes, streptomycin synthesis. Yes, you take the any name of the enzymes which is synthesized by the actinomycetes species, proteins, amino acids, wide range of the components synthesize this group actinomycetes, and this group able to synthesize extracellular or intracellular metal nanoparticles. Fungi is another group. Yes, eukaryotic cells, and they have the specific mechanism for synthesis of these components, including the yeast. That is wide range of the application of these microbial groups which is used for synthesis of these nanoparticles. We'll take some example. Yes, example, one or two examples of bacteria, actinomycetes, or it may be fungi. That is intracellular nanoparticle synthesis by a bacteria that use pseudomonas species and that form the silver mine. Yes, that particular species is isolated at specific temperatures, pH, and placed in the aqueous solution of silver nitrate solutions. And there may be by process of reduction that particular bacterial species, by the reduction of the silver ions, it is synthesized the silver nanoparticles within the periplasmic space of the bacteria, that is inside the cell wall. Yes, and that specific microscopy, we can observe that some of the particles present on the surface of the cell wall, but most of the particles are there that present inside the cell wall, and that is in the periplasmic space of the bacterium species. Yes, that these triangular nanoparticles that have the specific structure. There may be synthesis of some silver sulfide crystals also. Yes, that is because of silver particles react with the presence of some H2S, and they form specific nanoparticles that have the ceramic metal composites and that have the wide applications in optical and electrical properties. As such type of the nanoparticles we're going to synthesize by simple bacterial species. Again, actinomycetes is also source of nanoparticles. As we just discussed, that is, this is one of the best group of the microbes which is used for synthesis of antibiotics and enzymes. And here it is produce enzymatic reactions in the reduction of the metal. And it is able to synthesize highly stable nanoparticles, and that species is thermomonospora species. Ultimately, this name itself indicates that extremophilic species that grows, multiply at higher temperatures, higher pH. This thermomonospora species, and if you're going to expose to the chlorourate solutions, then pH 950 degrees centigrade, and they have the reduction of gold nanoparticles. And here it is observed that particular species synthesize these nanoparticles by extracellular in natures. By extracellular in natures, that is gold nanoparticles. Then again, some researchers, I think you may not, you may know, that is Afsar sir as well as Shastri sir from NCL, has performed some experiments, practically experiments, and they prove that yes, this biological source is one of the most important source for synthesis of nanoparticles. Yes, and this filamentous bacteria, actinomycetes, not only synthesize extracellular, it also synthesize intracellular nanoparticles, that by the gold nanoparticles, by using novel alkalo-tolerant actinomycetes rhidococcus species. Alkalo-tolerant that able to grow at higher pH or utilize the higher pH for growth and multiplications. Again, this is a rhodococcus species, and it is the type of the group of the actinomycetes. And very simple procedure we can use. It's not much complicated, simple procedure we can design for synthesis. That is we can grow in the specific flask, simple process. 100 ml you can use simple media. This actinomycetes species require just carbon nitrogen source growth and multiplications, as maltose, glucose, yeast extract medium, simple media we can use for growth and multiplications. And cultures are grown and continuous sucking by rotary shakers, 200 rpm, pH 7, 27 degree, 7 degree, 96 hours. This is a slow growing species again. This is a slow growing actinomycetes, not growing as like bacteria 24 hours. This is the filamentous group, spore forming group, require more time to growth and multiply, at least 2 to 3 days, as like fungi. Hence this group is called as filamentous fungi. Hence how the conditions as like fungi. Mycelia were separated by centrifugations, 5000 rpm, 20 minutes, washed up by the distilled water in sterile conditions, and then this harvested mycelia mass then resuspend with the chlorouric acid solutions. We can going to mix with the chlorouric acid solution on the rotary shaker, and there have the biofilm preparation, and that we're going to analyze it. Here it shows some of the diagram. Yes, first biomass, first a diagram by observation, it shows that simple mycelium of that species, simple mycelium of this species in the culture medium. But once you're going to expose to the chlorourate solution, they have completely changed the color. First, it is a colorless, but it is changed the color in the aqueous solutions. by the formation of these gold nanoparticles, not only by observations. As a science student, we not just by observations and will not believe easily by anyone without any experimental proofs. Perform the UV analysis. UV spectra recorded of this biofilm radiococcus. First, before this exposure, it shows straight line. There is no spectra. But second curve, it may observe that after the exposure to chloruric acid solutions, then it shows sharp peak, sharp spectra of that particular gold nanoparticles, and that confirm that nanoparticles form in this solution, and that easily observed by this spectral analysis. Again goes for transmission electron microscopy. Yes, that is intracellular component. First observe that externally some cell wall component, they have the smaller gold nanoparticles. But afterwards, scientists first believe that these particles are very smaller but present only on the surface of that mycelium by observations. But if you going to transmissions and internal structure, if it is going to observe that 70 to 75% nanoparticles present inside the cell wall, and that is in periplasmic space of that particular cell. As that indicate that synthesis is occur, that particles is designed is inside the cell wall and periplasmic space, they have the specific size of the nanoparticles. Yes, that is some examples of bacteria and filamentous bacteria which is used for synthesis of nanoparticles. Then another species, fungi, that is ultimately it is the eukaryotic cell, and this species shows some special importance in synthesis of nanoparticles. Simple deal with the laboratory. We know that easily grow, multiply, easily identify this species compared to the bacteria. And particularly this species secrete more amount of proteins and enzymes. And whatever nanoparticles synthesized by this species, that is more stable as compared to bacterial species. Yes, this because of that stability or that particular proteins present in microbial cell, that is the most important. They use this species for synthesis of nanoparticles. Then one of the species, verticillium species, intracellular synthesis, silver nanoparticles, and that have the specific size 25 nanometer, that may be synthesized by using this species. Yes, specific size by silver nitrate it may be used, and that metal ion formation of silver nanoparticles. They have specific mechanisms, that is trapping on the surface of the cell. They have the specific electrostatic interaction of the ions, that is silver have the positive charge. Ultimately, external cell, that particular enzyme synthesize, that have the carboxylic groups, that have the negative charge. And because of this specific interactions, charge interactions, electrostatic interactions, yes, there may be formation of these silver nanoparticles. Yes, and if it is the intracellular, we know that how this intracellular component we're going to extract from the cell. We can use ultrasound techniques, some detergent techniques, or some physical methods we're going to use to dissect the cell wall, and we can collect the internal component from the cell. We use different techniques for isolation of these nanoparticles. Then extracellular by the Fusarium oxysporium species, that is the again one of the fungal species. They synthesize extracellular gold nanoparticles or cadmium sulfates, extracellular synthesis. That may be based on the release of some enzymes, reductase enzymes. And this enzymatic reactions responsible to synthesize this specific nanoparticles, and that nanoparticles are more stable nanoparticles. Even it is going to expose this species species mycelium to the specific quantum dots, that is its CDSO4 solutions. We can cadmium sulfate quantum dots we can synthesize, and they have a lot of applications in different sources. Yes, that is synthesis of extracellular nanoparticles from fusarium oxysporium species. We can use wide range of the yeasts also, not only filamentous fungi. Yes, that is a candida species. We can use synthesize the semiconductors. Yes, cadmium sulfides, lead sulfides, some other components, other semiconductors. We can synthesize some yeast unicellular components, and one of the new yeast isolated yes, from our Agarkar Research Institute also, that may be able to synthesize extracellular silver nanoparticles, and that yeast given the name MKY3. Yes, that research laboratory of Agarkar proves that yeast not only synthesize intracellular, it is able to synthesize extracellular nanoparticles. Yes, by using the specific yeast cells. Yes, that is yeast also one of the best source we can use for synthesis of nanoparticles. Why only microbes? Why not other biological source? Even plants. Yes, there is a lot of examples of plant extracts. It may be root, it may be leaf, it may be seed, it may be peel. Yes, that particular extract of these plants, yes, we can isolate it, separate it. We can use specific technique for extraction, isolation. And if that particular extract expose this particular metal solutions, yes, it may be silver nitrate, chlorouric acids, or specific quantum dot synthesis. Yes, then that particular plant extract, they have the specific enzymes, and that may able to synthesize these nanoparticles. Yes, we can easily prove that, observe that by maybe UV absorptions, FTIR, or it may be zeta potential or electron microscopy. We can use to check these nanoparticles without any enzymes, without any chemical components. Yes, directly we can able to synthesize these nanoparticles from plant extract, and there may be a lot of references is there. We can use these plants for synthesis of nanoparticles. Yes, they have control the specific size. They able to capping capacities, because some of the components, some of the active metabolites that may be responsible to coat these particles and stabilize this. Yes, size morphology, specific size we can use, or we can synthesize this plant assisted nanoparticle synthesis. Then there is some important parameters in this process. Yes, in the plant synthesis, that is the reductions. Yes, that is, we use some contributors, that is biological components. Polyphenols are there, flavonoids are there. There may be enzymatics, proteins are there. Yes, these components by the process of reductions is converted into small metal ions to the nuclei. And we can easily observe by the specific FTR, or it may be color change. We're going to observe that, and then it may be able to synthesize specific size. Yes, control particle number, specific size and shape. It may be synthesized because that particular enzymes have specific groups, functional groups. Some pH sensitive chemistry is there that may be responsible to synthesize these nanoparticles, and these particles then it stabilize by using some stability agents. Then some proteins are there, polysaccharides are there, phytochemical residues are there that may responsible to alter the surface chemistry of the particles, and because of that, that reduce the aggregations. And if aggregation is reduced, that how the stable nanoparticles it may be synthesized. And then again, this biology, some of the have the biological activity, surface molecules that have the antimicrobial activity, antioxidant activities, that it may be combined with the specific drugs as anti-cancer activity, or it may give some catalytic response. Yes, that have the specific steps for production of the nanoparticles, enzymatic reductions, nucleations, capping to make the stabilizations, and then it may have some applications in the pharmaceuticals. Particle size and morphology. Yes, that is, this is the most important parameters. In the nanotechnology, how it is depends what type of the size it may be produced. What is the morphology of nano? Because finally action, finally use applications, it depends upon the size as well as morphology of that particular nano components. Again, it is depends upon type of cell. The wide range of the cell we use is from eukaryotic to prokaryotic to plants. Which cell is used? Yes, what is their component? What is their morphology? What is their nature? That's one parameters. The second, that is type of metal ions. Yes, which ions you're going to expose? Either silver, semiconductors, it is gold nanoparticles, cadmium sulfate, whatever, which nanoparticle component we're going to use. Then pH of the media, what type of the pH that we're able to, that cell it may be exposed, cell that maybe grows. Yes, then incubation times, total duration, it depends upon cell to cell. Composition of culture media, which type of the nutrients are used for growth and multiplication of the cell, which type of the carbon nitrogen source we use, and total biomass concentrations. This cell is spore carrying, non-spore carrying, filamentous, non-filamentous. Yes, what type of the growing characteristics of the cell is there? That's again one of the parameters. That is biomass concentrations plays the major role. These parameters decide your size of the particles as well as morphology of the component. Yes, then how it is going to use these nanoparticles? Yes, if once it is synthesized, yes, we are able to identify it. What components are there? That a lot of methods are there to identifications. What size is there? What composition is there? What morphology is there? Yes, based on this, that nanoparticles behaves in the tissues or in the cells in different way. Hence, before that, we have to study or identify these components. Yes, how this source may be differentiate, able to synthesize different nanoparticles, which is going to isolate it from the plant, going to isolate it from the microbes. Purity and safety of that components. Is this metal is toxic? What concentration is toxic? What is their sterility? What is their cytotoxicity? That how to analyze. Stability also their aggregations. Study the accelerator stability, storage content, protein absorptions. Yes, and then we are going to study the applications of that particular nanoparticles in different drug delivery systems. Yes, where it going to apply this nanoparticles, that is wide range of applications of the nanoparticles Yes, in different drug delivery systems, it is very difficult to talk all these things, difficult to go in details. Now, a area of the nanotechnology only. But this nanoparticle synthesized from microbial cells, it may have wide applications, which is used as antimicrobial, is used as antibiofilms, and they have specific metal oxides or plant capped nanoparticles we're going to use for this process, even for biosensing and diagnostics. The simple chips are prepared. That chipset may be coated for diagnosis. And if that particular chips have some coating agents and it may go to the specific site, it gives specific fluorescence, or if it is exposed to the disease, it easily shows, it easily identifies, it easily gives the signal. There's presence of specific site, presence of specific tumors. Such biosensing diagnostics, it may possible by using silver nanoparticles, gold nanoparticles, quantum dots. Yes, environmental treatments, that is again waste treatment, environmental treatments, and there is the plant biotechnology. Nanocarriers can cross assist the plant barriers. Yes, there may be use, yes, mesosiliconized nanoparticles, mesosiliconized nanoparticles, and there is the experimentally proves that, yes, yes, is it possible to transfer the DNA? It is possible to transfer the drug direct in the tissue animal cells, because animal cells don't have the cell wall, but it is very difficult to transfer in the plants. But experimentally it is proved that intact plant, intact leaves, yes, if you use such mesoporous functionalized silicon nanoparticles, that you are changing some nanoparticles by using specific techniques, easily possible to transfer the genetic component? Is it possible to transfer the drugs in the intact plant cells? And they are made possible to develop the new plants. As such applications, yes, is there to use these nanoparticles in plant cells. Have some applications shortly we're going to discuss. There's stents we use that have a lot of applications to avoid the blood clottings, yes, damage of the nerves or the damage of the blood vessels. But such stunts, it may be coated by the gene and antibodies, and it may be transported to the specific size. Yes, nano coatings that may be responsible to improve the drug loading, and that may be possible to release this drug to the particular sites. Yes, or it may be possible to coat the genetic materials as a DNAs. Yes, to deliver the cell, directly transfer the DNA to the cell, siRNAs, small interfering RNAs, which is responsible to degradation of the messenger RNA. It may be responsible to synthesize some toxic proteins. Yes, or such components we can coat to the stents, and it may be delivered in the particular sites. Yes, and they have specific localized effects. Yes, not only use as a heart attacks to avoid the clottings, but such component responsible to release the drugs as well as genetic components to the specific sites. Then exome powered neuropharmaceuticals, exome powered, yes, that is small molecules. Yes, there may be, we know that delivery of drug to the CNS, central nervous systems. It may be Alzheimer's disease, Parkinson disease, tumor, strokes, psychiatric disorders, that is, or CNS disorders. Glioblastoma, that is the brain tumor. And it is very difficult to treat such CNS disorder because that CNS have one barrier that we can call as blood-brain barrier. Yes, that is used for the protection of the brain, but we have the limitations to penetrate the drug to the brain for the pharmacological effects. Yes, these exosomes are natural nanocarriers. Yes, we use liposomes, artificially synthesized liposomes as polymers, using the polymers. But exosomes, which are used from the natural sources, these vesicles, natural sources, and which is used to nanocarriers, that is, we can able to, this particular natural nanocarriers have more biocompatibility, low immunogenicity, stability, and easily pass the blood-brain barrier.
Sir, sorry to interrupt. There's one question in the chat board. Which method used for DNA sequencing? Can you just, I think on this slide only is asking one question. Dr. Umesh, Professor Umesh Bansur sir is asking.
Which method?
Which method used for DNA sequencing? I don't know why. What is the relevance for that? But okay, otherwise we'll take it at the end.
At the end, we'll take the question.
Okay.
At the
sorry,
end,
sorry for interruption. You can continue. Sir.
Yes. All questions we'll take at the end. No problem, sir.
Yes, sir. Thank you.
That is the exosomes, the natural component we use as a vesicle to transfer the genetic component, or we can use to transfer the drugs for such type of the CNS disorders. Yes, that is the neurodegenerative stroke diseases, some specific therapy that is related to chemotherapeutics, brain tumors, glioblastoma. We can use such type of the vesicles that we can easily pass to the blood-brain barriers. And nowadays, we use the treatment that is direct applications to nose to brain drug delivery systems. That is to avoid the brain barrier, we will not go for other routes as IV routes or maybe oral routes. Yes, directly if you apply nose through this trigeminal nose, yes, direct drug reach to the brain and they have the specific effect, and that may be possible by using this system. Biohybrid micro or nano robots, yes, that may be used active delivery for wounds. Yes, that is magnetic based nanorobots for diabetic wound healing, as well as microphages. Yes, biohybrid micro robots, which is used for cancer therapies, that may be responsible. Yes, small nanorobots. Yes, nanotubes, we can design it that, and we can transfer the specific genetic components. Yes, combine these magnetic components, and they have able to penetrate, which is high rate. Yes, high rate penetrations, and they have the antioxidant actions, and they have the antibacterial effects also. Yes, biological cells or that bacteria itself acts as a carriers for the active tumor homings, deeper penetrations. Yes, and control the release that may possible. Yes, by using these nanorobots, which may synthesize. Yes, by using biohybrid techniques or biohybrid process. Then structural DNA nanotechnologies, that is therapeutic and immune engineering, 3D, 2D structures. We can enhance the drug loadings and how the specific applications, specific functionalized DNA tetrahedron structures that may able to synthesize small nanotubes, and that nanotubes we can easily penetrate, easily enter inside the cells. It may have the applications in drug delivery, vaccines, immunoengineering, tissue regenerations, diagnostics, or it may have the biosensor applications. Then theranostics and nanobiosensors that may be used imaging and treatments, that is identifications. Yes. Before treatment, we have to identify the specific cells. It has a specific infections, diagnosis. Yes. Before that, we can easily treat if it has that specific infection into the particular cell by using some biomarker detections, infection disease, cancer imaging. Yes, we can use specific treatment. Yes, diagnostic signals. Yes, nanobiosensors, which is used in antibodies, aptamers, enzymes, nucleic acids, and we can use specific systems for identifications of disease uh, before the treatments. That is the nanobiosensors and theranostics applications. Gene immune therapy nanostructures as a functional biological interfaces. That is nanofibers we can use for tissue engineering and regenerations. It may be used in the bones. Yes, some specific nanoparticle component transferred in the bone recreations that may be given in form of the gel, and after specific period it may be converted in the bone formation. This is called as tissue regenerations. Bone regenerations, it may possible by using the nanoparticle therapy. It may be used nanofibers that may be used gene deliveries. Yes, and cancer also. Yes, nowadays we know that gene therapy that may use wide range of the vectors. Probably we use viral vectors, and we know the limitation of the viral vectors, that in form of the immunology toxicity. Then researchers use non-viral vectors as the liposomes, other nanocarriers to transfer the genetic component or transfer the gene, but still have some limitation of the liposomal systems. Yes, that is what is the rate of transfer integrations. Yes, we can use such nanoparticle component that may coating with the gene and that able to transfer the gene in gene therapy. Yes, that how the specific applications of gene delivery systems. Yes, here example is there. That is liposomes for drug delivery systems. It is also a phospholipid vesicles. Wide range of the drugs, lipophilic, hydrophilic drugs, we can easily embed it, easily loaded in the liposome systems. Yes, in the diagram itself, it is given hydrophobic drugs, hydrophilic drugs, DNA genetic materials. We can easily coated this lipoidal layer, and we can able to transfer to the cell nucleus this genetic component, this vesicles, and some cell, that is some parental cell, we can isolate this cell membrane, natural cell membrane. Yes, and that cell membrane we can fuse with some nanocarrier components, and that may also easily use for drug delivery systems. Yes, then liposomes, same custom, these liposomes or gene therapy for cancer treatments. That is gene therapy, this liposome drug delivery, vesicular drug delivery systems we can use for treatment of the cancers, or lipid-based siRNA nanoparticles, which is used for oncogenes, immunopathway silencing to avoid the synthesis of some toxic proteins, some cationic liposome-based plasmid derivatives that have the specific charge that we can positive charge, that have the negative charge genetic component easily embedded inside it, and we can easily deliver it to the specific site. Then this nanotech components used in the plants also, plant regeneration and gene transfer. We can develop the complete new plants, hybrid plants, transgenic plants. Yes, as I take an example of mesoporous silicon nanoparticles, nanotubes. Yes, and how the expression, there may be coating some, not only genetic component, they have coating some chemicals also, some activators also. Once enter inside the plant cell, yes, they have uncapping the particular components, and because of uncapping that internal component, it is genetic component, it may be released and expressed in the genes. Yes, that is carbon nanotubes is also used, and we can able to develop wide range of the hybrid plants. Yes, transgenic plants, so we can say synthesize by using transfer of the gene by using this specific magnetic or silver or gold nanoparticles, and we can able to develop specific new plants. Yes, then generally connection of biological synthesis to drug delivery systems. We are use this all biological component. It may be plants that may be able to synthesize these nanoparticles, microbes which is able to synthesize nanoparticles as well as semiconductors, able to synthesize protein enzymes as the stabilizers. Simultaneously, we use liposomes or other magnetic nanoparticles, and they have specific applications in the drug delivery systems as antimicrobial agents, as the diagnostic, as the imaging, as a catalyst. Yes, and there may have some coating. Yes, with some specific drugs. Yes, anti-cancer drugs, their solubility may be enhanced. We can coating with some antibodies, these nanoparticles, to produce the active targeting to the particular sites. Yes, to deliver the or to treat the cancer by using the cancer gene therapy. It is maybe possible to transfer the genetic component to the different active cells. Yes, that is the application part in the pharmaceutical sciences. Then what is future of this nanotech? We can say, yes, still some of the nanotech component in the pharmaceuticals, vesicular systems, we routinely use in the drug delivery systems. But such new derived nanoparticles from plants and microbes is normal formulation, it may be used, but it is have some applications or it is some limitations, yes, to use with the drug delivery systems. Yes, and that how the better opportunity to go for the gene therapy if you go use the nano components. That is the future. Is it possible to deliver the drug proteins and peptides other than IV route? Still, it is very common route for protein and peptide IV routes, not stable by the oral. Yes, not possible to penetrate absorbs from the external transdermal route, or still there is not much success by mucosal route or nasal routes. Then this nanotech approach in future it is may useful for protein and peptide synthesis or their applications or their use in the drug delivery systems. It is personalized and individual medications. We always say, now doctor generally we ask what is your symptoms, or it may be doctor asked to check the your bloods and then find out what is the symptoms, what is the disease, and based on that it may be prescribed the medicine. But in future doctor may ask you what is your genetic sequence, gene sequence, and based on your gene mapping or gene sequence, it may possible to prescribe the medicines. And for that, is every individuals that different medicines it may be used, and they have the better opportunity in this area. Nano devices, nanobots. Yes, is this nano device we can synthesize nanobots? If you place in the IV routes, you place in the blood, it going to the particular site, it may be analyze the disease, it may analyze the cells which have the disease, which have the cancer, which detect that particular cells. We can design these nanobots, and that boats you can add some anticoagulant agents. Yes, and that it may be transferred. Yes, yes, in the IV routes, and that have a lot of applications in nano devices and nanobots. Still have some future applications, microbiology point of view. Yes, particularly in the bacteria and active myocytes, it is possible. Researchers tried some few strains. It's just as trial and error methods, not as per references. But bacteria, particularly in active myocytes cell, it is possible to strain improvements. And we know that strain modifications, yes, strain modification, development of the new strain, yes, they have the different wide another applications or which may able to synthesize specific size other nanoparticles. Even media modifications, wide sources of use as a nutrient for growth of the cell. If you're going to change the component, we have some reference. One cell produce the antibiotic, if we change the media composition, that cell not able to produce the antibiotic. Hence, media component also plays a major role for synthesis of proteins or active components. And we have to modify the media component, carbon and nitrogen sources that may able to synthesize different type of the nanoparticles. Other microbial groups we have used wide range of the groups. Still, there is a very primary research which is carried on the plants. Yes, we can use wide range of plant sources, and that nanoparticle synthesis we can try to some other metals. Yes, there's only specific metals that we use, or some other solution we can use able to synthesize these nanoparticles. And today we know that, yes, how much microbes you know. Yes, out of that, yes, whatever microbes you recorded, studied, that is only 4 to 5% microbes is known. Still, we are not able to isolate, separate wide range of the microbial cells that may have the different applications, wide application in the field. And we have to look for the isolations and separation of these microbes. Yes, that is from my side. Thank you very much for listening, and I am very happy if you have some questions, yes, for the audience. Yeah.
Thank you for your very insightful, very informative session, sir, as usual. So that shows reflections of your all the entire commitment to the profession. Thank you, sir. Now there's a question answer session from the audience. I think I can take first questions from the chart board. There are two questions I can see. Then I'll ask one by one, not an issue. So there's a question from Dr. Prakash Halami. He's asking how biotechnology can help as a next generation drug delivery. What are such specific example in this technology has been found more effective?
That we have already taken many examples.
Already I think you covered that in the session. Yeah.
Some the nanobots or it may use in gene therapies, that is may be used in the cancer treatments, or it may be particularly in the diagnosis of disease. That is also most important. And we already taken some examples, and it's used in this next generation drug delivery systems.
Very good.
Yes.
Yeah. So participant, you can unmute yourself if you want to ask the questions. You can unmute and you can ask. I think that is the most better option, I can say. Yeah. Yeah. Anyone have any questions? Please let me know. There's one question from Professor Umesh Bansode. Sir. Sir, which method used for DNA sequencing? Sir, can you ask the question directly? Because we did not understand exactly what correlation
Gene sequencing general method, that is PCR technology, it is maybe used. PCR nowadays is direct gene analysis. Old methods are there. We go for the PCR and then amplifications and then it may be analyzed. That same technique we use for the DNA sequencing.
Yeah.
Now it is easily by the PCR method, that is PCR technique is used.
Yes, great. I think there are good comments, responses. Very nice session, very informative session. Good. Thank you for all the participants for giving the comments on even YouTube. Also, a lot of comments are there. So very nice session, excellent session, informative session. I can see a lot of comments in the YouTube also. Thank you all the participants for your positive response. Definitely, yeah. I'll one questions from Sarika ma'am, sir. Which epigenetic tests are prominent for Alzheimer's study?
We can't such one test we can say. Yes, Alzheimer's, that wide range of tests we can use for, that is what stage Alzheimer's are there. Specifically, we can't say that is this particular test we can use for that particular.
Correct. Yes, I think there's a question, even response is very informative session, all very informative. Yeah, Yogesh sir, can you ask directly questions? No problem. You can unmute yourself, you can ask me. You can unmute yourself.
Hello.
Yeah, please, sir.
Sir, just wanted to ask, like, whatever the microorganism or the, especially the bacteria and fungal part, sir had explained, can we use the fermentation technology rather than the normal part so that again it can give us a byproduct as well as the nanoparticles?
That ultimately the fermentation technology means what? We can growing the cells only by using the media. Yes, similarly, here also we can grow the cells by using the media, and that biomass or biocomponent we use for nanoparticle synthesis. This is part of the fermentation only. We can't separate it. This fermentation process and this nanoparticle synthesis is part of the fermentation only.
Yeah, just wanted to ask, like, can we have something like a secondary metabolite as a product as well as the nanoparticles? So that means within the same fermentation technology, we can get the both the thing. One is nanoparticle, and second one the expected byproducts from that bacteria. That is what I just wanted.
No, your purpose should be one. Yes, if you
Okay.
want to have, if we want to synthesize bioactive molecules, that cell it may be different. Or your purpose is to synthesize only nanoparticles, that cell it may be different. That media components, we can optimize the media component, we can grow the cell, we can decide the fermentation time based on your active component. It may be primary or secondary metabolites. Their criteria is different. And if you're going to use this cell for the nanoparticle synthesis, then we design the criteria as per our nanoparticle synthesis requirement. All this, it is not so easy in microbiology. We know that all optimized conditions in fermentation process or this process also, we just taking the example, we referring this particular some articles, but this completely trial and error method. If you take the example of one bacteria, which nitrogen source, which carbon source is required for growth, it is not so easy. We have practically we have to use different sources, and from that we have to select it. Hence, our target is different. Based on the target, we have to optimize all the conditions. Simultaneously, both we can't do it.
Good. Very good, sir. Nicely
Okay.
answered,
Thank you,
and
sir.
very good area where we can explore. There's one questions from, I don't know the person name, but what is the role of future microneedle drug delivery in veterinary challenges? Is there any research group working on this area?
Exactly, I don't know which group is working, but micro needle, as human beings, they have applicability in targeting. Yes, similarly, that may be used in the animals also. Yes, we have to check veterinary colleges, Bombay Veterinary College, someone working.
Yes.
Yes.
Yeah, one more question from Professor Umesh. Can nanotechnology method used in genetic disorder? Already you have answered, I think, sir.
Yes.
Already there. Yeah. Great. Yes, I can see the questions already. Sir has answered almost all the questions, and thank you for all the comments, responses on YouTube, as well as these questions already we have taken. And sir has answered very nicely. So yeah, with this maybe, sir, I'll be maybe going with my vote of thanks. If any questions are there, sir is always there on your, maybe I can share the sir email ID. You can
Yes, yes, sir. Yes,
drop the question on email ID. Probably
yes,
sir will answer, not an issue. Okay. A
yes,
presentation, sir, some is asking presentations. I don't know, sir. They're
yes,
asking
yes.
you PPT. So yeah,
Yes, so we will give the PPT,
yeah,
no
yeah.
problem.
Sir will give the
Yes,
presentation and I'll email you on that. But
yes.
use it very wisely. Don't misuse that
Mm.
presentation. This is
Oh,
very,
yes.
yeah, sir has made a lot of efforts to prepare a presentation.
Mm.
One question from last question is coming, Dr. Prakash, sir. And how nano fertilizer are useful, sir? Asking question. Nano fertilizer.
Nano fertilizers that mainly in the plants. Yes, that related to, I think, plants, that is proper growth and multiplication of the plants, that we can use this nano fertilizers. Yes, they have the wide applications used in the plant biotechnology, and we can design or specific nanoparticle fertilizers by using specific techniques, and that may be used for plant growth promotion, plant integrations. Yes, that have the application in the plant biotechnologies.
Yes, sir. Thank
Yes.
you. Thank you, sir. So please accept our token of appreciation as a certificate, sir, from our Society of Microbiology, as well as from Chair President Dr. Arvind Deshmukh, sir, as a token of. We will be sharing this on your email also. Maybe.
Thank you,
This
sir.
month.
In
Yeah.
short, you have prepared the certificate also.
Yeah, yeah.
Yeah.
And I really express my sincere thanks, sir, for you accepting invitation on this busy schedule and giving very insightful and very informative and brainstorming session on nanotechnology, how it is really future, one of the future technology, futuristic technology. Now we are integrating with AI ML. Certainly, you are giving the right path to this young, scientists, budding pharmacists, so that definitely they will take this as a one of the node, and they can use your knowledge to update their skill, and they can also come up with some publication, some patent. I think that's an achievement from our side and your side also. Thank you once again, all the participants, for joining and giving responses. I request all the participants kindly fill the feedback form so that it will be really helpful for your future webinars. We are coming with a series of webinars from the sir, as well as more experts like Dr. Kokre sir. So I really express sincere thanks once again from Microbiology Society of India, Dr. Arvind Deshmukh sir as the president and as the chairman of National Microbiology Society, Pharmaceutical Microbiology Society. I express sincere thanks to all. Thank you, thank you. Have a good day and have a happy
Yes,
Sunday.
yes,
Yes, sir.
thank you, sir, very much.
Yes,
Thank
yes.
you. Yeah, yeah.
So can we have one photograph, please? Can you just put on the your camera for 2 minutes? We'll have one photograph. Just quick. I want to take photograph. Please, all of you just put on your camera if possible for just 1 second. I'll take one memories for all. Yeah, please. I'll just take it this. Great, good, good. I'll take it in gallery just a minute. Yeah, great. I can see the good faces over here. Great. Yeah, it is there on. Fantastic. Okay, okay. Let me take pictures. Okay, thank you all of you. And you can leave the meeting now. But fill the this feedback form. Okay. Thank you once again, Kokre sir.
Thank you, sir. Thank
Yeah,
you, sir.
yeah,
Yeah.
yeah. You
Thank
can
you.
leave it. Thank
Yes.
you. Bye bye.
Okay, sir.
Arvind sir, you are there? Dr. Arvind sir? Deshmukh sir? Yeah, I think sir also leave. Okay, no problem. Thank
Yes,
you.
sir.
Bye.
Thank you, sir.